Neuronal glutamate transporters control dopaminergic signaling and compulsive behavior. Genetic studies implicate SLC1A1, a gene encoding the neuronal glutamate transporter EAAC1 with OCD. Here's the gene. In the central nervous system, a synapse is a small gap at the end of a neuron that allows a signal to pass from one neuron to the next. Synapses are found where nerve cells connect with other nerve cells and can either be excitatory or inhibitory. Excitatory synapses are linked with glutamate ions. Glutamate receptors in synapses are classified into three types: metabotropic glutamate receptors and ionotropic glutamate receptors, which are further classified into GLU-A or AMPA receptors and GLU-N or NMDA receptors. EAAC1, which is a neuronal glutamate transporter, is abundantly expressed in the striatum, a brain region hyperactive in OCD. Our findings show that EAAC1 limits activation of metabotropic glutamate receptors in the striatum, and by doing so promotes D1 dopamine receptor expression. Targeted activation of signaling cascades coupled to metabotropic glutamate receptor in mice expressing EAAC1 reduces D1R expression and triggers repeated motor behaviors. Intracellular GQ signaling cascades coupled to metabotropic glutamate receptor activation are able to regulate both D1R expression and the phosphorylation state of DARPP32 rapidly. The functional role of EAAC1 differs across brain regions, and not only are there regional differences in the density of expression of EAAC1, but there are also regional differences in the distribution of other molecules susceptible to the activity of EAAC1. The findings that EAAC1 show reduced D1R expression is consistent with recent radiolabeling assays. The DARPP32 phosphoprotein is abundantly but not exclusively expressed in D1 emersons and is known to integrate glutamatergic and dopaminergic transmission in the striatum.